Now, I was very saddened to hear about Peter Jusic's death. We were colleagues together at the University of Oregon for 4 years, and I got to know him reasonably well, and he will be missed. So it is, well, not exactly an honor, but it's kind of a privilege to be the first speaker in a series that is dedicated to his memory. The title of the talk nominally is Time Frequency Auditory Representations in Bat Sonar, and the other title, which is the one I wrote before I realized I already provided the title, is How Do Auditory Mechanisms Explain Perception in Bat Sonar? And I want to say a word or so about the odd form of this title. The title is not Auditory Mechanisms of Bat Sonar. We know a lot about the auditory system. We have no idea how it or the brain work, but we know a lot about the brain, and we know a lot about the auditory system. And maybe someday, some fraction of what we know will tell us how it actually works. But at the moment, we're deeply ignorant and at the same time overwhelmed with facts. It's the knowledge that's the problem. All right? What I want to emphasize is not, we know something about the mechanisms the auditory system contains, and what I want to address is how those mechanisms can be brought to bear conceptually into understanding how the bat sonar works. That's the tone of this. I'm going to move very fast because echolocation and the auditory system of bats is mostly an unfamiliar topic. And if I'm right, there are people in this room who have a wide range of different kinds of interests, from psychology and studies of behavior in animals to auditory physiology to signal processing, where there's not a lot of overlap among these interests. And bat sonar is an interesting topic because it unites forcibly those kinds of knowledge and those kinds of interests. There's no way to talk about bat sonar without talking about signal processing. There's no way to talk about bat sonar without talking about the amazing things they can do with it. And there's no way to talk about it without discussing at least a little bit about how the auditory system manages to achieve it. I'm going to move very fast, and if you have a question, particularly if you have a question you think might affect other people's understanding of what I'm saying, make yourself known, all right? And the usual way to do it is stand up, shout and scream, do this sort of thing. Don't be timid or shy about it. But if it's a 3dB kind of question, leave it for later, all right? Just because there's a lot to cover, all right? Since I moved to Brown University, the people that have been involved, the usual suspects, so to speak, that have been involved in this work are listed here on the left, and the sources of funding for this work are listed here on the right. Of these, the Office of Naval Research is naturally the largest partner in the operation because the goal of this operation is to design and build a biomimetic sonar that can outperform man-made sonars underwater. And sad to say, the process of designing a system that will outperform may be hard, but getting any kind of outperformance is not all that difficult. Man-made underwater sonar systems perform miserably in comparison with biological systems, and we desperately need to close that gap. All right? Now, the principles, this is boilerplate, this is the kind of stuff you put in a grant application for someone to read if they're not interested in the substance of what you're doing. But you fill the grant application with paragraphs like this that give it perspective, all right? This is for the number 2 reviewer than others. The number 1 reviewer would read the grant proposal and instantly home in on what counts, the graphs of the data, not the boilerplate. But the basic point is this. When the bat emits a sonar sound, it hears the sound going out. And most, if not all, of the neurons in its auditory system discharge a single spike. Later, it gets an echo back at a delay of 3 milliseconds, 10 milliseconds, whatever, and most, if not all, neurons in the bat's auditory system respond to the echo. It's immediately clear that the most important thing is not which neurons respond and which neurons don't respond. It's when all these neurons respond. And this is an unfamiliar view of the way the brain works. We're used to the idea that perception arises from a kind of inventory of the features of a stimulus, and that what you perceive depends upon whether there's a lot of reds and a lot of greens and a lot of rough contours and a lot of over here to the left turned on at once. And if they all happen together, that's probably how the perception is grouped, all right? In the bat's auditory system, most of the neurons simply fire once to all the pulses and all the echoes. And the feature detector stuff The fact that some neurons respond to some sounds and not to others is at the margins of this process. So keep that in mind. What will count for creating the perception is when the neurons happen to produce their single spikes. All right? We study a bat called the big brown bat, common all over North America. Related bats are found all over the world in one guise or another. It's a bat with a wingspread in engineering units of a foot. Okay? It weighs in metric units about 25 grams. All right? Just so you know that there are different kinds of units. All right? If you put them together, you might miss Mars or something like that, but you shouldn't be able to have a problem with that. It's a small animal. It flies along emitting sonar sounds through its open mouth, right here. These sounds radiate virtually omnidirectionally in the frontal half of space. The bat doesn't produce a pencil beam of sound, and I'll show you data on that later. The ears point in such a way that the left ear receives sounds from this half of the world, and the right ear receives sounds from this half of the world. None of the tricks that you would use in radar or sonar of beam forming for the transmission or the reception are involved here. It's all brutal, fast signal processing that happens much later. That's why this is interesting. Now, they're insect-eating bats. The most commonly observed behavior is this. Now, what you saw is a bat videotaped with a thermal imaging camera that uses the bat's own body heat to generate the image. The sounds are from a bat detector, which demodulates the high-frequency transmissions of the bat and writes them on the audio track of the camcorder, which is recording the output of this hideously expensive infrared camera. The camera's thermal sensing element cools itself to 70 degrees Kelvin, and there are a number of interesting technical problems that go along with that, such as what happens when you're holding it and this 70-degree Kelvin node is located about this far from your ear. All right? And between you and it are two circuit boards and a very flimsy piece of plastic. It gets cold. All right? Listen to the sounds. The bat characteristically adapts the transmitted signals to the distance of the target when it's in a situation where there's one insect. Okay, one object in isolation from other objects. The insect is visible in this situation as a point. It's a June bug. This time of year in Rhode Island, May and June, these beetles are the prime food of the bats. Beetles of this size are warm-blooded. They sit in the grass and they warm up like this, and when they get their flight muscles nice and hot, they take off and fly. That's why we can see them with the camera. If it was a mosquito and it was a small bat, we wouldn't see it. It'd be pointless to show it. Most of what people talk about in echolocation is this single bat, single target pursuit sequence, and it's a nice model for an aerial interception process. Okay? The sequence of signals that the bats emit, first in total like this, slowed down. Okay. Now, those are not bat detector demodulations. Those are the actual ultrasonic signals slowed down, I hope, 16 times, although it might be 8 or 32. You know how that problem goes. Something gets played and then reproduced, and then it's played and reproduced, and by the time you actually get to the wave file, it's not entirely clear what the time axis is. That should be 16 times slower. Each signal is a chirp, multiple harmonic chirp, several milliseconds in duration. Early in the sequence, and this is a typical signal here, early in the pursuit, there are two strong downward sweeping harmonics. The collective frequencies are from here about 20 kilohertz to here about 100 kilohertz. All right? Towards the end of the sequence, this is the sound that the bat makes, slow down. Again, it's a frequency modulated signal. This low frequency point here is dropped down to about 15 or 16 kilohertz, but the upper end of the band is still in the region of 100, 110 kilohertz. Okay? The thing that's interesting about this signal that is unengineering-like from sonar points of view is the presence of the multiple harmonics in the sweeps. Okay? To a sonar engineer, 90% of the cost of the sonar is the low distortion transducer, and the rest of it is you buy something for signal processing off the shelf. All right? A lot of effort has gone into low distortion transducer design. The bat says the best transducer is a 50-cent transducer that produces 50%, 100% second harmonic distortion, or worse. In this case over here, roughly equal apportionment of energy into the first 3 or 4 or 5 harmonics, and that's a lot cheaper to make. And the higher harmonics are free bandwidth. That's the first lesson from this. It's very important to think of ways of getting the bandwidth up without raising the cost of the system. This is the basic operating space for a BATS sonar system. All right? The BATS working in the range of 20 to 100 kilohertz face the very severe atmospheric absorption effects at frequencies above about 25, 30 kilohertz. At 100 kilohertz, atmospheric absorption approaches 10 decibels per meter, which means if the sound goes through 10 meters of space, it's attenuated by 100 decibels. The BAT transmits a sound with an amplitude of 100 to 110 dB right here. If the object reflecting the sound is a perfect sphere with no target losses, that's going to be near the threshold hearing of the BAT only as far away as 7 meters. So in principle, a small insect-sized object will be invisible if it's as far away as 10 meters. So much of the system that the BAT uses is organized around a search space of the order of 5 or 6 meters. This curve here shows the BAT's threshold of hearing for several different targets and echoes to show essentially the operating space. The space that the BAT uses sonar in acoustically is in this sort of curved triangular space right here, above the green line and below the ideal blue line, where the echoes from real objects would be located. They can see insect-sized objects as far away as about 5 meters. All right? A single isolated target produces a single echo for each transmission. And perceptual segregation of one echo from another, say the echo from an insect versus the echo from a tree off in the distance, this is not a problem because these echoes are removed to very different points in time. All right? However, that's what happens when you have that aerial interception. Now, BATs, however, often initiate aerial interceptions where the insect and the BAT are over here like this, but during the course of the pursuit, the insect moves toward the vegetation. Many insects hear the ultrasonic signals of the BATs and take evasive action. And one common example of the evasive action is to dive to the ground or to spiral into the vegetation. Nevertheless, the BATs can come quite close to the vegetation and make a capture. And that is the summary of what most people would have thought was the limit of this catching system. Sure, the BAT can catch the insect if it's near the vegetation, but only if it's acquired the target relatively far away. I'll just glue this. This, by the way, is a slightly lower quality infrared camera here, and a little cheaper. All right, it's handheld. All right. Okay. It's much less appreciated that bats also will dive right into the vegetation and pull the beetles right off the leaves. Presumably listening to the buzzing sounds of the beetles, their wings hit the leaves and produce an ultrasonic signature up to about 20 to 25 kilohertz with a fundamental at the wing beat rate. And you can hear this, the bats can hear it, and you can actually attract bats to fake buzzers. The bat goes right into the vegetation, and when you get the angle right, you can see the bat pushing the leaves apart as it moves in there. It clearly can cope with highly cluttered environments. That's not the sort of thing a man-made radar or sonar system would like to encounter. It would be a very difficult task because the leaves are just as reflective as the insect. All right, one more time, listen to the sounds. When the bat is in the vegetation, the sounds are weaker. And they're not weaker because the vegetation hides the sounds. We've put microphones right up in the middle of that. What happens is the bat is aware or has learned that in these conditions the beetles can hear it coming. And so the bat either shuts off the transmissions or reduces their strength to avoid alerting the beetles. Sometimes in situations like this, when you run the video and look very closely, you see beetles streaming away from the area trying to escape. All right? Okay, bats on the, I apologize, some of these videos are not the world's best infrared quality. This is not National Geographic stuff, but there's a limit to what you want to spend on the cameras. All right, bats also have dog fights with each other. Okay, those bats are doing that in total darkness. They're flying at 3 to 5 meters per second. The whole process is guided by hearing. The chasing bat is presumably using sonar to follow the chasee. The chasee is using its sonar to avoid crashing into things. And if you will notice, just watch. It actually dives to the ground to try to shake off the pursuer. And it's presumably also listening to the sounds of the bat chasing it to guide its escape. So it's a very complicated process. You had a question?
I was wondering about the dive to the ground. I was wondering whether they're chasing an opposite.
No, they're chasing each other. They do this a lot. Sometimes they spend more time chasing each other than feeding on insects. And in a situation like this, it's clearly not territorial or driving other bats away from the food, because there are 3 bats, and there must be about 50 metric tons of beetles in the tree next to them. So there's... They're probably playing, and they're also probably related to each other, because they come from the same colony. So there's any number of explanations, and none of which is any more or less plausible than another. We just don't know what we saw. Other than that, it is possible, at least for bats, to use this kind of system for mutual pursuit. Imagine 2 aircraft solely guided by active and passive radio emissions producing a chase. That's not easy. All right? They also do this. They're drinking, and they're also chasing the mosquitoes that are hatching from the surface of the water. So the drinking, you can see when they touch the water and it produces a ripple. Bats drink on the wing by dropping the lower jaw and tongue into the water and splashing some in the mouth. And they're catching the insects that are coming up off the water at the same time. And there are half a dozen bats within a few meters of each other at any given moment. So you can hear a lot of bat sounds in the background. Also, and this is much worse, they do this. The duty cycle of bat sound approaches 100%. There are 50 to 100 bats within 5 meters of the center of this cattle watering tank. There's just no way we can figure out how this happens. These bats fly around like this for 2 hours every night, and they've been doing it for hundreds and hundreds of years at sites just like this in the Southwest. There are Indian stories of these bats accumulating over water at night. They do this every night. They don't bump into each other. They get fat while they're flying around, and they don't exhibit the characteristic behavior that you might think that if their sonar is primarily for obstacle avoidance as well as for catching insects, you don't see very many instances in which a bat will abruptly change course to avoid a collision. Instead, what you see are multiple, long, loosely correlated paths. That means that these bats, in effect, are dancing with each other. Each bat knows enough about where the bats are near it that they can move in coordination. That means they have very sophisticated real-time images capable of mixing active sonar echoes with the passive emissions of the other objects, which means it's like human hearing. We can listen to stuff and it's trivial. They do this stuff and it's trivial. Figuring out how to build something to do it is very hard because we're not sure what we saw. We're not entirely sure what's actually happening here other than the phenomenology. One of the things we do is we go out and we videotape them doing this sort of thing. That's mostly to show you what people look like with one of these cameras. All right. We'll stop in a moment. And to remind you, notice that the dark glasses are opaque. These wavelengths don't go through glass, so it's not a glass lens. It's a germanium lens, and that's why the camera costs a lot of money. There are a lot of problems with these long-wave infrared signals and trying to image from them. All right? What we do is we attempt to reconstruct their flight paths from three-dimensional reconstructions using 2 synchronized video cameras, and that gets us into yet other problems, because if you do a 3-dimensional reconstruction of bats flying in a place, you need to have objects that are part of a frame of reference for this. And that turns out to be best attacked as a problem in small-scale topographic mapping. And to do a small-scale topographic map right, you have to go back 50 years into the history of civil engineering to find the tools for it. Otherwise, you buy some $50,000 GPS device and you fall in the water with it, okay? And whereas these transits, okay, you can take one of these things, lob it across the room, pick it up, make some adjustments, and go on at 20-second angle measurements just as before. They're very resistant to damage, and it's very easy to use these things for topographic mapping, stadia measurements from distance and stuff. If any of you have done any civil engineering work, you know how much a vanished art this is. Okay? And we've had to learn about this stuff. This particular transit here was made when I was in high school. And it was abandoned 20 years ago by the Michigan Highway Department. And then it turned up on eBay last summer. That's how we got it. All right? Okay? All right? Now, most bats hunt insects. But quite a few species in the New World only, these are not found in the Old World fruit-eating bats, flying foxes, have night vision, a really good, highly sensitive photo accumulation system. In the New World, fruit-eating bats are sonar guided. This is a fig tree. It's not well shown up. It's a little bit better on this thing here. This is a fig tree full of figs. This glowing object here is a giant termite nest. It's up in the tree. It's in Belize. All right, and what you're going to see is fruit-eating bats diving into this tree to land on the fruits. They're very silent. They make very weak sonar sounds. They're hard to hear. There's a bat sitting there picking a fig off a cluster. They typically, I want to watch this again while I talk here. They typically dive right into the tree to find the figs. All right, now this tree is covered with leaves. None of the figs is visible from outside the tree except during the bright daylight when some of the colors of the riper figs show up. But this is the middle of the night when visual cues are useless. They find the figs as acoustic targets. We think because the spherical figs are omnidirectional radiators, and when a bat is passing into the vegetation, the leaves speckle and flash on and off, but the figs themselves stay on all the time, and that's how they can dive into this vegetation and find them. The reason their sonar sounds are nearly indetectable is that instead of being 110 dB, they're 65 dB at the frequency range they use, because hovering above the tree are large carnivorous bats looking for the fruit-eating bats. And if they radiate sounds too loud, they get eaten. Now, this is what's interesting about it. When bats hunt for insects or deal with objects in vegetation, the problem is they get multiple overlapping echoes. And the existence of one object versus another comes to be a problem in segregating the acoustic stream so you can tell where the various reflected replicas of the incident sound are hiding. All right? The transmitted signal that the bat uses is essentially omnidirectional in the frontal third of the world. It's plus or minus about 3 dB at 45 degrees left and right at most frequencies. At 22 kilohertz, which is the search frequency, it's plus or minus about, 4 or 5 dB at 90 degrees. So they've actually found a way of beating some rules about aperture design and radiation of sound. They're radiating much better than their aperture should allow. In the horizontal dimension, the beam is pretty broad also. It's pointed down because these experiments measure horizontal in terms of the eye-nostril plane of the bat like this. That's how you make the measurements. And under those conditions, the mouth is pointing down. The flying bat, of course, has its eye-nostril plane tilted up, and this thing would be nearly at the horizontal search level. All right, the ears are the receiving antennas for the auditory system, one pointing to the left, one pointing to the right. This is the left ear radiation pattern for different frequencies, receiving pattern. Essentially, it receives most sounds coming from the left and not much in the way of sounds coming from the right. The complementary pattern is true for the right ear. Neither the transmitted sound nor the receiving beams narrow. These sounds, essentially if you have 4 or 5 objects in front of the bat out here in space, those objects will return echoes to the bat's cochlea with roughly equal amplitudes from different positions in space. So the only differences that the bat will have to look at the locations of these objects will be differences in time and phase at the 2 ears. the amplitude cues, in particular, in the range of plus or minus 15 degrees, which is where the bat mostly keeps a target during tracking, are virtually non-existent. So it becomes a problem in time and phase measurements for wideband signals using receivers that are only 14 millimeters apart. All right, this is the vertical beam. It's even more complicated. The vertical beam is very, very complicated in its appearance across frequencies because I've selected widely spaced frequencies, 5 kilohertz sampling, frequency sampling interval here. And what we're seeing is an interaction of this rather wide sampling interval with the directional characteristics of the external ear. The external ear has a large receiving surface called the pinna and a much smaller reflecting structure in the front called the tragus. This is the pinna right here, and this is the tragus right here, this blade-shaped structure. Sound entering the ear reverberates back and forth in this narrowing waveguide, and there are actually diffraction pattern-like ridges on the ear which help the reflective properties of it to make it non-specular. This reverberation generates an elevation-dependent time delay in the sound reaching the eardrum, which, because the sounds are only offset by a few tens of microseconds, generates an interference pattern in the spectrum of the sound, which is elevation-dependent. This is an acoustic measurement, frequency from 20 to 80 kilohertz, elevation plus or minus 60 degrees. There's a spectral notch which moves from about 25 kilohertz at 40 degrees down, moves to, oh here, about 50 kilohertz at 20, 25 degrees up, and then it vanishes. That's the spectral notch. It's a little bit warpy because different bats are holding the ears at slightly different attitudes when you do the experiment. It's kind of hard, all right? The best way to do this is to train the bat behaviorally, to fixate its head at a loudspeaker and then present sounds from loudspeakers at different elevations and probe for the location of the notch in the spectrum caused by the interference effect. And when you do that, you get a nice rational plot showing the second order, the interference notch for the time delay for this reverberation. That's the cue the bat uses to determine elevation. The ears are tilted outward like this, so that this elevation cue for each ear is not up and down like this, it's really inclined, so the bat has a pair of cylindrical coordinates set about 45 or 50 degrees apart like this. So by comparing the notch in the left ear and the right ear, it has an independent system for determining azimuth and an overlap system for determining elevation, and it's quite clever. All right. The main point is that the lack of sharp directional gain in this system means that if a bat wants to distinguish between an object here and an object here, if they are about at the same distance, resolves into a problem of detecting very small differences in the arrival time of what otherwise are closely spaced overlapping echoes. The external ear feeds sounds into the cochlea of the bat, which operates like any mammalian cochlea. The signal is passed along a dispersive delay line, which mimics what looks like a bunch of parallel bandpass filters. To pursue that analogy much further than saying that gets weak because it stopped acting like that. But to a first approximation, if this is a chirp from 100 kilohertz down to 20 kilohertz, it gets broken up into segments by bandpass filters, which look at, say, an 80 kilohertz, a 70, a 50, and a 30 kilohertz segment of the sweep. The Q values of these filters are in the range of 5, 6, 7. So the integration characteristics of this impulse, you get 5 or 6 or 7 rings as the signal goes through, and then the signal is halfway rectified and smoothed, like this. And it's these envelopes that trigger nerve discharges in the bat's auditory nerve. The halfway rectification permits this system to encode phase and be coherent, which is interesting. The critical question is, if the question becomes, you have this smoothed envelope and you have some ripple on it which reflects the phase structure of the signal passing through the bandpass filter. We need to know what the frequency cutoff is for that bandpass filter. It's been measured in cats, in monkeys, in rabbits, in gerbils, and several other animals at 1.5 kilohertz. It's never been measured in bats. All right, there are a number of reasons why it hasn't, but the short of it is the physiologists who work on this are chicken to take on that really very technically difficult experiment, but it's an important one to do. Our behavioral tests and modeling show that this low frequency cutoff is probably around 10 kilohertz, an order of magnitude higher than it is in cats, for example. But then cats don't fly. All right. Now, this spectrogram-like representation of parallel bandpass outputs produces this sort of pattern here, where if this is the transmitted signal, now I've removed from these figures the complexity of the multiple harmonics in the FM sweep, because once you start talking about a signal with 2 or 3 harmonics in this time frequency representation, you can start tripping over different pieces of the harmonics. The main point of bringing the harmonics in is to show you that most time frequency signal processing methods do not work here. For example, if you have a signal that has 2 harmonics and you use a Wigner-Will distribution to describe its time frequency characteristics, it falls apart because you have a signal with 2 frequencies at any given moment in time. And the Wigner-Will distribution doesn't like that, so it starts throwing new frequencies in all over the place as the cross products. Most time frequency representations fail to cope with real multiple harmonic signals, and these kind of short-term filtered, bandpass filtered methods are better. The transmitted signal, the returning echo. A bunch of nerve impulses generated in model auditory nerve fibers at different frequency channels trace the instantaneous frequency of the FM sweep. There are small differences in the thresholds for these firings, and the Schmidt triggers essentially for the nerve discharges, so you get differences in the pattern for the pulse and the echo. As the amplitude of the echo changes by 1 decibel, these things slide back and forth on the average by about 15 microseconds, which is the slope of the excitation function going into, as I say, what amounts to the Schmidt trigger. I say what amounts to because it's actually quite complicated. And we have 2 echoes separated in time by more than the width of the integration time of these filters, which is shown graphically by this envelope and is in fact plus or minus about 300 microseconds, which goes with the low-pass filter and with the Q of the tuning curve. You get independent discharge patterns representing both objects. The transmitted signal, echo A and echo B. It's easy to see in this representation where the insect is and where the tree is, for example. All right? The problem is, what happens when the 2 echoes become so close together that their frequency sweeps arrive inside the integration time for this short-term spectral history, or for the filters, or for the spectrogram, however you want to call it. What happens is that the signals merge into a single signal with an interference scalloped spectrum, and thus produce this very complicated discharge pattern here, which does not betray, at least in an obvious way, the fact that there are 2 separate reflecting surfaces hiding in there. Instead, you get 1 surface plus an interference pattern. All right. Let's see if this will work. Oh, this is boilerplate. Let's just, okay. You imagine now, if the bat measures the delay of an echo by measuring the time of occurrence of a nerve impulse at a particular frequency in the transmitted signal, the broadcast, and compares that with the time of occurrence of a nerve discharge arriving later to the echo, these delay measurements are used to compute an estimate of absolute distance. All right? And as long as you don't get spectral interference distorting this pattern, the picture of delay is reasonably accurate. But if the 2 echoes overlap like this so that there are interference notches in the signal, then the measurements of distance at each frequency get complicated by the shifts in latency caused by changes in amplitude, the Schmidt trigger, the amplitude effect. And so you wind up with a distorted picture of the position of the object in space. All right? Worse, this is the equivalent of a kind of spectrogram cross-correlation process. This spectrogram to this one here with a little bit of dispersion here. It's all done frequency A to frequency A, frequency B to frequency B. What the bat actually does is it freely discards rationality and simply compares all frequencies in the transmitted signal with all frequencies in the echo. This range of dispersion covers at least 30 or 40% of the band, and it may be as high as 100% connectivity. We don't know for sure. These tilted comparisons of time frequency points are a kind of bat's way of doing higher order spectral processing on the signals. But from the point of view of target ranging, they represent a problem. These lengths of these arrows all measure the distance to the target. And it's a useful piece of information until the bat changes the duration of the sound. And then all of a sudden, all of these diagonal lines have different numerical values than before, and they no longer become useful indices of the distance to the target. Instead, they become confounding. When bats are chasing insects in the open, they make sounds that change in duration, go chirp, 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 chirp. And the distortions caused by these cross-product delay calculations don't disturb the bat's ability to reach out with a precision of plus or minus a couple of centimeters to grab the insect. The bat usually scoops the insect up in its tail and then puts it in its mouth. That process doesn't disturb the delay measurements enough to make that impossible. But it makes it very hard if you're dealing with an insect in vegetation, because you have no idea where the insect or the vegetation are located, really. It's too smeared. When the bat is chasing an insect or trying to find an object in vegetation, it doesn't change the duration of the sounds as it goes closer. It doesn't go chirp, 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 chirp. It goes chirp, 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 chirp. It makes a rapid series of sounds of all the same duration, so that these sloping lines here are not shifting from one transmission echo pair to the next and creating unwanted distortions. They become part of the system for doing a deconvolution. All right, now we go to that. Echo detection experiments reveal that the integration time of echoes, behaviorally now, not in terms of those spectrogram filters, behaviorally is plus or minus about 300, 350 microseconds. That is to say, if you put an echo here and ask the bat to detect it, and put other echoes coming in like this, they will interfere with that detection process according to that curve there. You will get lots of detection errors when the interfering echo arrives at the same moment as the test echo. That plots the shape of this integration time function. All right? And you can show recording from nerve fibers. This happens to be a multiple unit recording from the nucleus of the lateral lemniscus, but you can show by a variety of methods if this is the transmitted signal being presented to the bat. It's awake, sitting there listening to sounds, and you're recording from the auditory system, and this is an echo. As the second sound, the echo gets closer and closer to the first, the existence of a readily detected response to the second signal vanishes, begins vanishing when you get down in the millisecond region here, and the shapes of these functions stop changing when you get down to about 300 or 400 microseconds. That's just a measure, a demonstration physiologically of the integration time measured behaviorally here. It's the same fundamental procedure. However, notwithstanding the long integration time, if you give the bat a pair of echoes in a range or delay measurement task, two echoes, only 100 microseconds apart, the bat will tell you there are two echoes and give you an estimate of each delay separately, which means it has deconvolved those echoes that overlap inside the 300 microsecond integration time and, in effect, read the peak and notch pattern along the spectrogram and transformed it entirely into the time domain. Separate experiments showed that there's no variance left over in the frequency domain when this is done. Whatever information is on the time frequency surface has all been moved onto the time scale by the time the bat perceives it. There isn't any spectral quality to the target independent of the range information contained in the echo. All right. The method we use in these experiments, I'll just show you this. One more, and I'll show what's happening here. Okay, what happens is the bat is trained to sit on this Y-shaped platform and broadcast sonar sounds into B and K microphones, one here and one over here. The bat sounds, the electrical signals are digitized at about 1 megahertz, delayed in a digital buffer system, and then reconstituted as analog signals at a delay of, say, 3 milliseconds, or 3.1, or 3.2 milliseconds, and fed back to the bat from a loudspeaker located here. The microphone and the speaker are about 20 centimeters away, and the electronic delays simulate objects at distances of half a meter or whatever you want. And you essentially train the bat to respond to these stimuli, to tell you whether there are two echoes present or one, and you move probe echoes around to show that they actually are perceiving the delay of the second of the two overlapping echoes. A lot of things of this kind. How small a jitter can the bat detect from one transmission echo pair to the next, for example. Psychophysical tests, the details of which I can go into later.
The bat doesn't have wings.
What?
The bat doesn't have wings there.
The bat has wings. It just walks on its... When a bat walks, it furls the wings and walks on its thumbs. A lot of bats walk. They locomote by getting around that way on the ground, looking for insects or for water. Yeah, they don't fly away.
And their acoustic perception when walking and flying is expected to be the same.
Of Who knows? Since you can't do the experiment when the animal's flying, it's a useless question. You know, it's in the sense that, I don't mean it's a bad question, I mean, but it's a pointless question to try to answer, because the effort involved in training a bat in a choice while it's flying vastly exceeds what you're going to find out. I mean, if it's the same, that's good. If it's different, then what do you do? All you can do is, you take the measurements that you get. This is where it's engineering. It's an animal, but it's basically engineering. When you do this experiment and you ask the bat to detect whether echoes are jittering delay versus stationary, and you change the size of that jitter interval, you find bats very easily can detect jitters of 40 or 50 microseconds, or 10 or 20 microseconds, but jitters of 0 microseconds, of course, are not detectable because there aren't any, and jitters of the order of 30 to 35 microseconds are hard to detect too, all right, for reasons that I'll get into in a moment. This is a basic result, and it traces a kind of time axis or a-scope image. Imagine the bat is over there, target ranging this way, and there's a point target right here. This shows the shape of the image of that point target as having a prime centrum where the target is located and a side lobe 30 or 35 microseconds later. Okay? And it's shown by the fact that the bat makes more errors at that point in the discrimination task. All right? We initially thought, oh, this is great. This is the envelope of the cross-correlation function of the signal. This is the central peak. This is the first side lobe, because that's roughly the period of the first harmonic of that FM suite. That means they see cross-correlation images. Well, it could be better in sonar or radar. That's what you do with sonar and radar. You figure out ways to get cross-correlation images, and then it's max filtering. We thought, oh, this is great stuff. So we did an experiment, all right, in which we trained the bats to detect jitter with 0-degree echo phase changes. That's this here, and it's the blue curve here, or a situation where we jittered the phase while we're jittering the delay from 0 to 180 degrees. And what we've got is this. If this is the cross-correlation function, this is the underside of the cross-correlation function below the 0 line. The bats actually were perceiving the phase delay of the frequencies in this signal, and because it's a frequency sweep, talking about phase and group delays gets complicated because of the really instantaneous frequencies. The bat sees the dispersion of the instantaneous frequencies in the signal caused by the phase inversion, which is very interesting. That means it's a coherent receiver, or it can be. All right? In addition, when the bat is asked in these jitter experiments to detect echoes that are 100 microseconds apart, they can easily tell you, but two echoes simultaneously 100 microseconds apart, they can assign independent delay values to those two. That's a resolution experiment. When the bat does a resolution experiment, what it has to do is find interference notches caused in the echo by the time separation of the overlapping echoes, and then somehow compute its way from the frequencies of the peaks or notches to the corresponding time estimate. On this axis, on this pile here, you have behavioral results for a single echo with no 0 microsecond delay separation, if you will, 10 microsecond, 20 microsecond, and 30 microsecond separation. The bat's ability to make delay estimates tracks the actual delays embedded in these overlapping signals, which means it can do the deconvolution because it's well inside the integration time. All right? When you do the experiments on a small fine scale, you see on the fine time scale, these curves have two peaks in them, like this, until the echo delay gets down to about 2 microseconds. And then when the delay separation reaches 2 microseconds, the bat no longer perceives that there are two delays present, it perceives only one. All right? The signals the bat uses are in the range of 20 to 100 kilohertz. The period at 100 kilohertz is 10 microseconds. The bat is working down in the range of 1/5 the period of the signals for fine resolution, which is a good demonstration of what used to be called in the radar literature hyper-resolution. There was a long argument about whether it was possible. And the outcome was that it is possible, but not easy, especially in real time. All right. More, if this business of the bat having this little side peak here, if the bat is capable of resolving echoes as closely spaced as 30 or 20 or 10 microseconds, this side peak here may not be the side peak in a cross-correlation image. It might be the consequence of something causing a secondary echo to occur to the bat. The sound that enters the ear canal bounces around between the parts of the pinna and tragus and arrives 30 or 40 microseconds later at the eardrum after the primary reflection. So we wondered whether this side peak here might actually be the bat sensing the time reverberation of its own ears and its own antenna. So we did the experiment of moving the loudspeaker, and sure enough, it turns out that this side peak is the bat getting the external ear's frequency response caught up in the deconvolution so that it creates additional time estimates in the image. All right. All right, at this point, I think maybe we should go to some physiology. All right. Any questions? How much time do we have? 10 minutes? Excellent. We'll do 10 minutes, all right? We could go on and on and on about those tests of performance, but I think I made the point that they're good enough to want to figure out how they do it, all right? The bat's auditory system consists of a cochlea, which contains a bunch of bandpass filters, so as a superficial model, lots of parallel nerve fibers leading into the brain along the auditory nerve from the receptor elements in the cochlea, and then a network of neurons that form an ascending cascade. The auditory nerve, the cochlear nucleus, the nucleus of the lateral lemniscus, the inferior colliculus, the medial geniculate, and the auditory cortex. These Latin-based words are biologists' or neuroanatomists' attempt to confuse you, to keep you from doing neuroanatomy and taking their jobs. Just as your use of mathematical symbols prevents biologists from going out and doing engineering, okay? It's very effective, all right? In this case, the problem is they're irrational in the sense that they don't mean anything. They just have to do with what the things looked like 100 years ago under a microscope. So the inferior colliculus is the back bump, all right? The medial geniculate is the middle body, which has the geniculate-like shape to the cell structure. And on. The cochlear nucleus is the first agglomeration of cells after the cochlea, on and on. This cascade of neural processing stages adds a millisecond or so of time delay as signals propagate up to the auditory cortex. All right? And here's a picture of what the responses look like if you present the bat with a sound at different frequencies at time 0. The cochlear nucleus, which is the earliest stage of the bat's auditory system from which recordings can easily be made, neurons respond to different frequencies of the sound, which is this axis right here, not showing up very well. That's the vertical or three-dimensional axis is frequency. This is low, around 20 kilohertz. This is high, around 100 kilohertz. The neurons fire once at a delay of 2 or 3 or 4 milliseconds. Okay? They pass their information synaptically to neurons in the nucleus of the lateral lemniscus, which fire a millisecond or two later, once, at delays of 3, 4, 5, 6, 7, 8 milliseconds. They pass information to the inferior colliculus, whose cells fire once, but produce this hugely complex pattern such that some neurons, when they fire, they'll wait 30 or 40 milliseconds before they actually exhibit their response. These cells here stretch the neural representation of the transmitted signal out to cover the time over which the echo will come back. So it essentially stores the shape of the spectrogram of the transmitted signal for comparison with echoes. I'll show you a better picture here. The top is the cochlear nucleus. I'll use the arrow here. This is probably easier. Neurons in the cochlear nucleus respond, and their responses are over with before the echo returns. Inferior colliculus neurons, however, start responding, but continue to respond for 30 or 40 milliseconds, covering the time window over which echoes can be expected to return. We'll come to these in a moment. The initial step, the cochlear nucleus, versus this inferior colliculus, which in effect can be thought of as a system of delay lines. When an echo occurs at a particular delay, here, this is a delay of what? I think we said it at 10 milliseconds. That's right, a couple of meters. The cochlear nucleus produces a second volley of neural responses to the echo. But it's not possible for the cochlear nucleus to compare the broadcast and the echo because the cochlear nucleus has stopped responding to the first sound before the second sound happens. Because all these cells fire only once for each sound, They can't do anything when they're not firing. They can't carry or compare information or execute computations. In the inferior colliculus, however, the responses to the echo begin happening well before the responses to the transmitted signal, the green ones, have ceased. And it's in this area of overlap between the green and the red dots that the computations in the nervous system for sonar take place. The auditory cortex, through the geniculate, looks at this pattern, and it makes a comparison between one red dot and one green dot if they are superimposed in time. The auditory cortex contains neurons that are time coincidence detectors for comparing pairs of cells in this green-red pattern here. One particular auditory cortex neuron might respond to a pulse and an echo at a delay of 5 milliseconds. Some other one will respond to a delay of 8 milliseconds, or 12 milliseconds, or 20 milliseconds. The auditory cortex segregates its responses to echoes by the time delay separation according to the patterns of coincidences that occur in this thing here. If I took the red dots and moved them over this way by delaying the echo, a different pattern of coincidences would occur and different cortical neurons would produce the output. These cortical neurons, now let's go back here. I want to just show you this dynamically. This is a static picture of what really is a system of dynamic delay lines. We have a pulse, pattern of responses propagating like this, and then an echo and a pattern of responses propagating like this. And I want you to see what happens if you animate the physiological data, as though it were recorded from a bat flying, for example. That rapid movement is it. The coincidence detectors look at, let's freeze it for a moment, any dots in green on this, in this column, and in this column, green are fair game for a coincidence comparison. There will be a neuron in the cortex that gets an input from this cell and this cell. And if the time delay of the echo is right, those two cells will fire at the same moment and in turn fire the cortical coincidence detector, which creates a coarse A-scope display. This gives you some idea how fast it is, and this is slowed down. The whole thing is over within 40 real milliseconds. All right. Let's go on here because I want to show you something else. This gets interesting. All right. we're going to go to this and then stop, okay? This is a very complicated system which uses dirty math, all right? It's the sort of thing which you would in high school would have, someone shows you a geometry proof that's quick and dirty, where at some point somebody takes a ruler and measures the side of the triangle, for example, a quick, dirty approach to the problem. Here you have, let's go back to this. This is a picture of the red dots, the responses to the echo in the inferior colliculus delay line system. It looks fairly random. It looks pretty disorganized. Is this a manual focus here? Let's see what happens. Yes. No, that's a size. Let's not screw with the focus. I'll do it differently here. Okay? The main point is that seemingly random distribution of dots conceals time structure. If you look at a histogram of that distribution up there, and great care was taken to be sure that the conditions for this dot here were the same as this dot experimentally, except the frequency of the stimulus, so that we don't have to worry about a lot of noise this way or smearing in the data. It's very hard to do this experiment right, but when you do it right, you generate a couple of hundred points like this, and this is a histogram of this pattern of discharges, latencies over frequency. If you look at it, there's some fine structure to it like that. The histogram is not a random distribution of points. It's organized, or at least pseudo-organized. If you take the AC signal riding on top of that up there and subject it to Kepstral analysis, it turns out that, we'll go back to this, that these dots here are really organized as though there's an initial burst of activity that is random, but then that pattern of random activity is repeated, embedded in this mess with a periodicity of the order of 5 milliseconds. This is a picture of the spectrum of the dots and the analysis of the spectrum to determine the time separation, which works out to be 5.5 milliseconds. And there are about 3.5 or 4 cycles of time processing in this thing. What's happening in the inferior colliculus is that neurons that respond early in this sequence send their information on up to the auditory cortex, which responds. Many neurons in the auditory cortex project directly down to the inferior colliculus, producing new responses in the inferior colliculus. The time delay for this up and down cycle is about 5 milliseconds. We think that most of the red dots over here, the ones at relatively long latencies or long delays, are generated by this recurrent process, and that's how they appear in these pictures. This is the recurrent process time dynamics here. That the delay line system the bat uses for measuring long delays is coupled to already having measured those delays. So the possibility exists for the bat to make a measurement and then essentially to make a repeated measurement on the same measurement rather than an independent measurement, so that the statistical properties of this measuring process start to get really complicated. I'm going to stop here because I think that this is a good place to stop and see if you have any questions. If some of the questions relate to some of the other slides, we'll switch it back on, but let's just stop. Thank you. You notice they're not answers to anything. They're just devices to generate more questions or more experiments, if you will. Yeah?
So earlier you talked about auditory cortex making delay computations.
Right.
And you said that those were independent of frequency.
Right.
In other words, I interpreted this to mean...
Right. Essentially, any combination of frequency you can find being used in these calculations.
I see.
Yeah.
Okay, so maybe this is a simplification, but a neuron in the auditory cortex has a positive input from a red neuron lower down and a green neuron
higher
those
up.
aren't necessarily paired.
Exactly. They're very often the red neuron may be at 30 kilohertz and the green one may be at 50.
Right. So the question is, are the neurons in auditory cortex that do this interchangeable? And as you implied,
we don't know. The basic part, the auditory cortex, just like the rest of the auditory system, has a frequency or topographic tonotopic axis.
yes.
When you get to the auditory cortex and you're looking at a neuron, it's going to be located in a place on the auditory cortex tuned to a particular frequency.
Tuned to a particular delay?
A particular frequency.
A particular
frequency. The delays are randomly scattered all over, like raisins in pudding, we think, that kind of thing. The question is, you're at a particular neuron, you're at a particular frequency point in the brain. Which frequency is it? The frequency in the pulse or the frequency in the echo? The higher one or the lower one? We don't know. That's a tough question. We do not know that. That set of experiments is to be done next.
You implied nonetheless that some information was lost in the process, and I was wondering how you know that.
I'll go over that question again.
You implied that some information was lost because of the randomness of the pairing.
Oh, okay. The main problem is, okay, all of those time delays, the diagonal ones and the horizontal ones,
yes.
make sense as long as the signal's duration is not changed. If the bat keeps the same sonar signal, that matrix of calculations will correctly produce an estimate of delay. All you need to do is know what the frequency differences are in the sweep. In other words, if you know the shape of the spectrogram and you know the cross value, the cross products, you're home free.
Okay.
But if you change the signal, then you throw the whole computation up in the air and let it fall on the floor, and you have to pick it all about and start it over again. The bats change the duration of the signals on a continuous scale.
If you know that there is such a change in the duration of the signal, why can't you predict?
You might say one obvious thing here is the bat listens to the sound coming going out. So why not simply set up a new set of there are a bunch of neurons that appear sensitive to the local slope of the sweep. Why not simply set up a new set of rules for conducting the pairing? Cumbersome, but perfectly capable of being done, because one of the things you would not accuse the auditory system of being is efficient in terms of use of units. There are millions of these things in there, okay? So that's a cumbersome but computationally very good solution, very likely one of the things that happens, all right? A major problem is that we have evidence that the bat is aware of this problem, the limitation. When you ask the bat, sitting like this, it's moving around, is this object closer or is this object? That one's closer. You go, that one's closer. And it'll give you that judgment plus or minus 2 or 3 centimeters, okay? And while doing so, it will make sounds that vary in duration from about 1 millisecond to 2.5 milliseconds without much system. It'll go chirp, 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 and that's closer than that, okay? And that's all the money is. It has to tell which one is further than the other, but it's moving around. And there's not much it can do to stabilize its body better than a centimeter or two, because when it makes a sonar sound, it goes and it moves a lot, okay? What if we change the experiment so that instead of going do, 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 we change it so the bat goes duh, it gets one echo, duh, it gets the next echo, and the time delay is being given to the bat before it can move very far. So it's going chirp, 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 chirp. It's making, we're giving it a jitter rather than an absolute difference from left to right. When you ask the bat to perform that task, it no longer goes chirp, 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 chirp. It goes chirp, 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 chirp. They're all the same. And you can overlay the spectrograms, all right? And that suggests a type of coherence that works in the spectrogram but wouldn't work in the original time series signal. These signals have the same spectrograms, but presumably random starting phases. So if you take the time series waveforms and you overlap them, you discover they're smeared. But if you take the instantaneous frequency curves, and that turns out to be harder than it looks like because you have multiple harmonics, you can't use the Hilbert transform, it just explodes in your face when you have 2 harmonics or 4. It picks a harmonic, often something that isn't there. The spectrograms overlie in the sense that each instantaneous frequency point is nearer than one period of that frequency to the point in the other signal. So it's a different definition of coherence that the bat may be using to allow it to see the phase shifts. Now, this is speculation, but the bats do this, and we need to find a way to explain it, okay? That's a good point, a good point. There are lots of corrections the bat can make because, of course, it knows perfectly well what the signal was. It just made it itself. It chose that signal from its repertoire. It made it. It heard the signal with essentially 1,000 signal-to-noise ratio, so it can do whatever it wants. The echo comes back with a signal-noise ratio of 10 or 20 or 30 dB, and for the most part, that's good enough for these experiments. If you actually do the classic signal-to-noise experiment, at a signal-to-noise ratio of 36 decibels, the bat is capable of detecting on a single ping basis a 55 nanosecond delay change. In fact, they detect about 40 nanoseconds, but they use 2 or 3 signals. So it's just spot on to an ideal receiver operating on these signals. The problem is, how does the nervous system operate in the range of a few tens of nanoseconds? The bats regard 40, 50 nanoseconds as trivially easy to detect. If you use a correlation processor, you can find that, but 40 or 50 nanoseconds is a tiny fraction of the width of the central peak of a correlation function. And so a matched filter actually is working a lot harder than the bat seems to be. Of course, no physiologist in his right mind will believe or will accept the idea that the bat can do a 40 nanosecond discrimination. I have several grant reviews to prove it. Okay, any questions? Any further ones? It's a great engineering system, it really is. Yeah.
So is the reason we don't have artificial systems that perform as well as the bat because we lack understanding about what the bat does, or because we lack the technology to implement it, or both?
Okay, good question, good question. I think the primary situation is technological historical. Right? The use of delay lines for different frequencies was first introduced into radar in the 1950s at Bell Labs with chirp radar systems. At the time, no system existed for analog to digital conversion of waveforms of this type. And so what you would do is you take a chirp signal and feed it into a bunch of parallel bandpass filters with different delay characteristics that would compress the signal into its autocorrelation function, or for the pulse and the echo, the cross-correlation function. And for a decade or so, this engineering instrument looked a lot like the way the auditory system works. But at that point, what happened is the radar signals would be demodulated to an intermediate band, sampled digitally, and operated in using digital signal processing devices. Once you digitally sample a signal by the conventional time versus 10-bit or 12-bit sampling regime, you wind up with a frozen block of data, which you then have to go back to later and do cross-correlation with recursive moves. You take the signal, you delay it, you multiply each point by its friend, you delay it again, you multiply, you add, and you've done an awful lot of time-wasting stuff, which in a computer looks fast. Okay, but the computer itself is running like mad to do this stuff. And there's stuff going forward and backward in time all over. This original approach of using parallel bandpass descriptions of the frequency sweep and feeding them into a delay line creates an entirely feed-forward computational scheme whose output is a cross-correlation function. But by a system that didn't have to go backward in time to do it, because these dots here store the information for future calculations. This system can execute cross correlations, but it does them on the time frequency surface rather than on the original time waveform. When you think of the cochlea in engineering terms, the actual answer to your question, it is a different analog to digital domain. Rather than 10 or 12 or 14 bits by points in time, it's 1 bit by many, many points in time frequency. And it's a very different regime, which is much more dispersed and more amenable to fast calculations, but requires proportionally more computational elements to do it. So instead of having, 16-bit words being spread through 128 word arrays and operated upon over in serial events in time, you have thousands and thousands of 1-bit lines that go off in different time directions and then meet for calculation for computations later. It's a different strategy, very different strategy. And it's a technological, historical thing. DSP technology is so good for what we're doing, it's hard to see that other analog to digital regimes exist, radically different ones like this.
But there's no fundamental reason why we can't implement such a system.
There is a hidden dirty problem here, and this has to do with the bandpass filters in the cochlea. They're not passive operating devices, they're active, with the capability of boosting their own gain within the duration of a single cycle by as much as 50 decibels or so. So there's a lot of creative use of instability in this system about which we don't have much knowledge. They're hard, they're nonlinear in multiple ways, they're very hard to model, and they're active. And it's very, very difficult to get a handle on this kind of system. Okay? There are people here who are Johns Hopkins who are as good as anyone in the world trying to do this, and it's hard. Further question? Yeah.
So does that use the same system for listening to lower frequency sounds, or is there a parallel apparatus, or
Okay.
does it just extend down that level?
Okay, our bats hear sounds from 10 to 110 kilohertz. That encompasses echolocation. It encompasses their social communication sounds, which are mostly in the range of 10 to 50 kilohertz, and often, in a developmental sense, are derivations of biosonar signals. They don't hear below 10 kilohertz or 8 kilohertz. They're deaf. They're stone deaf. One experiment we did seemed to show they could hear around 1 or 2 kilohertz, but we were never able to satisfy ourselves that the loudspeaker wasn't producing distortion. That was a tough problem. Other species of bats sometimes hear to low frequencies depending upon what their social or environmental needs are. There's a bat that hunts scorpions in the desert, and in addition to using sonar to avoid crashing into things, it listens to the sound of the sand grains being kicked aside by the scorpion, which is a low frequency random noise-like event, and they home in on those sounds. So it's a tough question to answer. For our bat, they only hear in 10 to 100 kilohertz band. Other bats may be different. Okay? Thank you. Was this satisfactory?